So our next speaker is Mimi Ho from Columbia University, and the title of her talk is Visualizing the Translational Choreography of Malaria Parasites.
Thank you so much, Martini, and thank you to Peter and Prakash and Audrey so much for putting together this amazing symposium and inviting me to speak here. I'm really excited about this talk because this is the first time I'm going to be presenting new data from our lab, so very excited to be here. So, sorry? Is this better? Yes? Okay. So I don't think that this audience needs an introduction to malaria, but this slide I'm just kind of using to demonstrate that we're mostly working on kind of the asexual blood stages of the parasite. Is the laser pointer not working?
There it is.
Okay. So in our lab, we use structural biology to kind of understand the host-pathogen interactions, specifically in the asexual blood stages. And so before I dive into that, I just want to kind of explain to you that as a structural biologist, this particular parasite is very challenging to work on, particularly for us, because the genome is so aggregation prone. So there's a lot of highly charged, low complexity regions that are littered throughout the entire proteome, and that makes these proteins extremely difficult to express in recombinant systems, which is what we traditionally as structural biologists like to do, right? So it's very easy to make E. coli or yeast kind of grow up a ton of your proteins so that you can use it for structural studies. But in this case, most of the proteins that we're interested in don't express well in recombinant systems. And so what we've had to do in order to do structural biology on this really fascinating parasite is find ways around this problem. And so in my lab, how we've, during my PhD and then now in my lab, how we've chosen to get around this problem is basically by going after structures directly from, like purified, enriched from the actual parasites themselves. And this is made possible by this technique called cryo-electron microscopy. So now it's been long enough since the resolution revolution, I'm sure lots of you are aware that the CryoEM has kind of really broken open the doors on allowing the structural characterization of proteins that were previously really not possible unless you could get tons and tons of that protein purified, usually from recombinant systems. So just to kind of tell you a little bit about the two techniques that we use. So it comes in two flavors, and the goal of both of these techniques, cryo-electron microscopy, single particle cryo-electron microscopy, and cryo-electron tomography, is basically, at the end of the day, to get a three-dimensional reconstruction at high resolution of your protein complex of interest. And our goal in doing that is basically, we need to get different views of your complex from many, many different angles so that we can come up with a 3D shape, right? And so there's two basic ways to do that. So the first way is called single particle cryo-electron microscopy, where we purify our protein out of cells. We take lots of cells, we purify our protein out, we freeze those on a grid, and then we put it in the microscope, and you take lots and lots of images, and you get millions of particles of your protein that are randomly suspended in the ice, and you get a lot of your different views, all represented from your particles being in different orientations in your micrographs. And so then you can classify those and then use those to kind of get these different views or 2D class averages, and those you can use to back protect to get your, to back calculate to your 3D volume that gave rise to those 2D projections. So that's kind of the work that you're probably more familiar with. There's been a lot of really cool structures that have come out very recently, specifically from malaria parasites using this technique. So there's this beautiful MSP1 structure that was done recombinantly by Misha Khrushchev. This, obviously, you guys are probably aware of the PFCRT structure, also done recombinantly from the lab of David Fiddick, ROPH complex structure, as well as the RH5 Psi-RPA Ripper complex from Alan Cowman's group down in Australia. And then, of course, there's my personal favorite, PTEX. So the thing, obviously, these structures are super cool. We learn a lot from them. But the thing that's really frustrating about them is that we don't get a picture like this, right? So we don't get the cellular context. We get just the view of the protein by itself. And you don't get any of that cellular context that tells you all of the cool things about what that protein is doing, right? So I think kind of this new angle in my lab is kind of born out of the frustration of going and giving talks about PTEX and having people ask me questions about like, how are proteins refolded once they exit the other side? Or how does PTEX recognize which proteins it should be unfolding? And I had to answer all of those questions with, I don't know, because unfortunately, we don't have the cellular context to tell us the answers to those questions. And so that's kind of really what motivated this new area, or kind of new area of research in our lab, now that I started my own lab, which is to do cellular tomography. So this is how tomography works, cryo-electron tomography. Again, you're trying to get different views of your object. But in this case, instead of having thousands or millions of different copies of the same object with different views in a static image, what we're doing is we're taking a single object and we're tilting it. So we take an image at 0 tilt, we tilt 3 degrees, take another image, and so we get images of the same object from many, many, many different angles, and that's how you get all of your different views, and you're able to reconstruct those into kind of a 3D volume that contains that particle, right? And so what does this look like when we actually move into cells and not cartoons? So this is what we're doing in our lab now to kind of get at some of these questions, is basically we're directly taking parasite-infected red blood cells. We're not breaking them open anymore, so we preserve that cellular context. We freeze them on the electron microscopy grid, and then we used a special microscope called a focus ion beam scanning electron microscope to ablate the cell mass above and below, the cell material above and below our area of interest. So this is, you're basically blasting away or vaporizing your material kind of one atom at a time. And so there are no cutting artifacts, so it's perfectly pristine. And because you've kind of vitrified, directly frozen the parasite, there's no fixation artifacts, no dehydration artifacts from fixing. So it's really as close to native as you can get. We take them out of the incubator, we walk across the hallway, and we directly freeze them. And so we basically thin down to this very thin section called the lamella, which is about 200 nanometers in thickness. And from there, then we take it out of this microscope and we walk across the hallway, and we're able to put it into the transmission electron microscope and take tilt series. So what that actually looks like, so here we have, we usually will grow up, we'll endogenously tag if we want to, if we need a fluorescence tag, and then we grow synchronized culture, isolate those parasites, and then we freeze them on this manual plunger at the bench in our lab. So this is what our manual plunger looks like. So here you can see that's the EM grid. The Sharpie's kind of just there for scale. And this here is the liquid ethane that we use to kind of plunge freeze to vitrify our cells so quickly that you don't have time to form crystalline ice, which would damage all of the cells, all the membranes, right? So we are putting it into the ethane so that it freezes so quickly that the ice crystals don't form and they don't damage your cell. And so the liquid ethane is cooled down by liquid nitrogen, right? And so what we do, so David in our lab has just applied kind of harvested parasites to the grid, and now he's attaching the tweezers to the manual plunger, and then he's going to take a piece of blotting paper and wick away the vast majority of the sample, leaving a very thin layer of cells, which then, if the video plays, he will then plunge down into the liquid ethane. So from this moment forward, that grid never leaves the liquid nitrogen ever again until we are done imaging it and we throw it out. So it has to stay under liquid nitrogen at all times from here on forward. So what we do at this point is we pick it up, we put the grid in a box inside liquid nitrogen, we put it into Dewar, get on the subway, and go down to the microscope. And so here, this is the special microscope that I was talking about, the focus ion beam scanning electron microscope. So this is what it looks like. This is my students, Mezi and Jing. They're cooling down the microscope and loading the sample into the scope. And so once you get inside, this is what the scope looks like. So this here is the electron beam, and this here is the ion beam. And so we're going to basically be imaging using the scanning electron beam from the top, so perpendicular to the surface of the grid. And then the ion beam comes in very, very close to parallel to the surface of the grid. And that's what we're going to be using to ablate our material. So once you get inside, this is what it looks like. So this is kind of the top view here. Over here is from the electron beam. And this is kind of the view from the ion beam, which is very close to parallel to the grid. And it's really nice that we can image with the ion beam at the same time as we're using the ion beam to ablate material, which is cool. So you can see here, we have really nice kind of layer of infected cells here. And we've selected a couple of cells that we're interested in here in the middle. This is not the high resolution image, but if you zoom in a little bit, you can sometimes actually see they look like an egg, basically like a fried egg. So you can see the red blood cell around it, and you can see a tiny little bump in the middle. That's the parasite. And so we're aiming basically for these cells right here. So we've got these landmarks to kind of help remind us at the lower angle where we're going. And so then what we do is basically set our boxes, and we use that ion beam to mill and we get thinner and thinner and thinner. And by this point, it's the end of the day, we've been here for hours, and we are literally sitting at the edges of our seats because you're down to the point where it's under 200 nanometers, and you're desperately hoping that it doesn't break, right? So if you manage to survive all of that, your lamella survives, and not all of them do. You've got this very, very hair-thin kind of layer here. It's 150 nanometers. This is what it looks like from the top, so you can see that it's so thin that all of the contrast has disappeared, and you can almost see through it. And so then what we do at that point is we take it out of that microscope, still under liquid nitrogen, and we put it into a dewar and walk it across the hallway, and then we put it into our transmission electron microscope. And when you put it in, this is what you see. So you can see here, this gorgeous lamella. You have a bunch of red blood cells here. You can see clearly these are the uninfected red blood cells, so they're completely flat. It's just hemoglobin. There's nothing going on there. And then if you zoom in here, you can see these gorgeous digestive vacuoles. And if you zoom in further, you can actually see all of this beautiful detail of the parasite living inside this red blood cell. So here, this is the uninfected red blood cell here with the red cell membrane. And here, this tiny little divot right here, you can see that's a knob. So this is the first time we ever saw this. Is super cool. We must probably, the strain that we're using is not super knobby, because I was kind of expecting to see knobs everywhere, but we don't actually see that many. We don't catch them that often, but this one is really kind of one of our favorites because we managed to catch this. So if you look closely here, this is the digestive vacuole. So you can see there's a beautiful digestive vacuole here. Here are the human zoom crystals. We seem to have caught a vesicle inside the digestive vacuole, which is cool. So perhaps maybe from a vesicle fusing in, because it's double membrane covered with the cytostome. And then when you get inside, I assume one membrane fuses with the membrane of the digestive vacuole, but the other one stays intact and must get broken down somehow. So here, next to it, you can't see super well, but this has multiple layers. That's an apicoplast. So it's kind of pushed up against the digestive vacuole. And here, if you look really, really closely, there's two very faint lines running next to each other here. That's the parasitophorous vacuole and the plasma membrane of the parasite. And all of this stippling that you see, very bright, very light stippling, that's all individual protein complexes inside the cell, intact as it was when we froze the cell, which the first time that we saw this, we were completely blown away. So as you can see here, as other people have mentioned, as Dan mentioned in his talk, there is a lot of hemoglobin going on, which means for us, for a long time when we first started doing this, I thought we were doing something wrong because our contrast was quite poor. For this is, I mean, compared to other systems, our contrast is very poor, and I thought maybe there's high molecular crowding, and then someone mentioned, you have all of that heme, and there may be labile heme around, maybe that's contributing to this like high background in your images. And so it turns out that it's true, we are getting down to the correct thinness. You should have better contrast, but I think that the background scattering from all of that iron is really kind of contributing to high noise in our images, which is unfortunate, but we deal with it. So this is already super cool. I mean, when we're at the microscope, we really just pour over the lamella, but the goal here is obviously to get to high resolution. And so what we do is we zoom in even further. So this is kind of like the field of view for one tilt series. So we're taking this at 1.7 angstrom per pixel. And so we zoom in here to a really, really high magnification, and this is where we're gonna take our tilt series, right? So we go in and we tilt from -60 to +60. And then when you reconstruct that into a 3D volume, so this is, for those of you who do confocal, this is like going through the Z-stacks, right? The Z-slices in your Z-stacks. So this is basically going through the Z-slices of this reconstructed tomogram. So this is a 3D volume at that place where we took the tilt series. And so now you can see with much better resolution or much higher detail that those red cell membranes, the knob here, the vacuolar and parasite plasma membrane, as well as all of these protein complexes. So then we do a lot of filtering and deconvolving to improve the contrast of our images further, and then we can use a neural network. We can train a neural network to recognize or segment out all of the cellular features, all of the ultrastructural features in the cell, as well as these protein complexes. And that allows us to get a nice three-dimensional colored reconstruction of the ultrastructure inside the cell. And the reason this is important is obviously because this is way easier to interpret than squinting sideways at these really, really low contrast images that are all grayscale, right? So this is already pretty cool, but if you look in at each one of these little golden potatoes is a ribosome, which is really neat. So you're looking at ribosomes literally as they exist inside the cell. They're undisturbed. But for us, I'm a single particle person, and so I'm used to seeing side chain level resolution, so these potatoes are a little bit disappointing, right? So what we do next is we actually box out, we pick out every single one of these little little potatoes, and we do something called subtomogram averaging, where we can bring them all together, we align them, and we average them to boost the signal to noise ratio so that we get higher contrast, which means higher resolution. And when you do that, you get something that looks like this. So this is a 3.7 angstrom reconstruction of the ribosome as it exists inside the cell. So this is the actual structure of these ribosomes as they exist inside the cell. And so you can see here all of the RNA helices. You can see on some of these, on these alpha helices, you can see the bumps that tell you those are side chains. So you can actually see amino acids on, like amino acid resolution detail on these reconstructions. So then obviously once we did this once, we were like, okay, let's go in and do it across the life cycle stages. So we did it for we went across and tried to do it for all of the different life cycle stages. Some of them are more challenging to work on than others. But here we've managed to do a high resolution reconstruction from trophozoites, and then also here on the bottom from merozoites as well. The merozoites are more challenging because they're tiny, and also there's way less ribosomes per merozoite, so you need a lot more of them. But here you can see, again, some really beautiful segmentation of this merozoite here. The rope shoes are my favorite. They're really cool looking. So now that we've done that, we've gone through and basically managed to get reconstructions from the different life cycle stages. The schizonts are the runs of the litter because they're the hardest for us to work on. We haven't developed this method quite as well yet, so we don't have a lot of particles yet, but we're working on gathering that data. And so the cool thing that you can do after this, so it's not just a cool picture, what you can do after this is that you knew, right, you have the XYZ coordinates of where every single one of these ribosomes were in your original 3D volume. And so what you can do then is you can take each of these ribosomes, the high resolution structure, and you can map it back into the original tomogram and then show it with the segmentation. And so now you can see all of those ribosomes within the cellular context, right? So you can actually really get back at the original question that we were trying to ask and the reason why we started on this crazy venture about 2 years ago. So you can see here, all of the ribosomes are mapped back. You can see, exactly where they are, the different ribosomes. You can see, some of them are interacting with the membranes. You can see where they are distributed in the cell. And so this, I think, was kind of the culmination of quite a lot of work in the lab, but we're very, very excited about this particular movie. So having been able to do this, then the next big question that we had is, okay, well, what can we do with it? Is it possible to actually visualize the molecular mechanism of a drug inside the cell? Because the whole point, yes, we want to be able to see structures of proteins inside the cell, but it'd be really cool if we could actually see what the molecular effects of a drug would be inside the cell, right? And so to do this, we kind of just set this challenge for ourselves, and we wanted to see whether we would do this. So because the ribosomes are so prominent, there's so many of them in the cell, they're kind of a really easy system to work with. And so luckily, one of the top drugs in the clinic right now is actually a drug that is known to be an inhibitor of the ribosome. So it's an inhibitor of elongation factor 2. It's known kind of through resistance mutations to be binding to EEF2, but it's not known, the molecular mechanism is not known, right? And so basically what we wanted to do is see whether we could kind of learn more about the molecular mechanism of this drug inside the cell. Because right now, kind of the limitations are we can learn a lot about the mechanism of a drug in vitro through biochemical or kind of in vitro assays, or we have kind of gym-sustained looking at phenotypes, but this is not at high resolution, right? And so what we would like to be able to do is combine those two things and be able to see high resolution detail inside the cell. So what we did first is we went through and we did a kind of a time course, a 48-hour time course where we took kind of blood smears every 4 hours, a little bit more frequently at the beginning. And so we went across and we were looking kind of at the course of what happened. So here you can see here's the control parasites going across 48 hours. You can see they develop into really beautiful schizonts here and then reinvade around here. And so in the drug-treated parasites, you can see that they basically stall out at early trophs and then they just persist like that for a really long time and then eventually just become pycnotic and die. And so it looks like there's not a lot going on here. And so when we were trying to choose which things to go after, the first thing we wanted to see is, okay, first of all, can we see higher resolution? Can we see more detail than what's going on in these smears to tell us kind of ultrastructurally what's going on with this drug? And so the first thing we looked at is we selected a couple of time points. So for us, based on the Nature paper that described this, the drug is active in culture. They see that it's inhibiting translation within the first 40 minutes. So they incubate the parasites with the drug for 40 minutes and they can see inhibition of translation. So we thought, oh, okay, 1 hour and 3 hours, probably a good way to go. If you wait too long, maybe you won't see the effects anymore. And so this is a control parasite at 3 hours. This is a drug-treated parasite at 3 hours. And you can see with some details, so I think that the organization of the cell is definitely a little bit messed up. The most obvious one is that the food vacuole seems really kind of smashed and condensed down and dehydrated a little bit. And to help us to see that better, we went in and collected tilt series all over, basically papered this entire cell with tilt series, and were able to fill in kind of much higher resolution. I know that it's not obvious from here, but if I were to zoom in on one of these and let it fill up the entire screen, we see much better resolution for every single one of these tilt series. And that allowed us to do this ultrastructural segmentation. And we're working on the ribosomes for this now too. So you can see here the rough ER, you can see a beautiful digestive vacuole. It's nice and big and round with hemozoan crystals kind of in the middle, and a lot of nice empty space around them. This here we think is probably the mitochondria and maybe an apicoblast next to it. And so compare that with our drug-treated parasite, and you can see here, this is the drug-treated parasite, and when you zoom in, again, with Ultrastructure, you can see here the digestive vacuole seems to be kind of condensed down. It seems like it's maybe blebbing off and making like a second digestive vacuole. And so this is kind of really cool, but then the thing that we wanted to do next is obviously see what are the ribosomes doing kind of between these two states. And so then ensued about 1.5 years of building up an insanely, for us, huge data set. So this occupies, I think, like hundreds and hundreds of terabytes. So just the data alone takes up hundreds of terabytes. And so we've gone through and basically collected tilt series. We've milled, tens to hundreds of cells across different life cycle stages, drugged and not drugged trophozoites. And then collected tilt series and picked particles out of them. And we ended up with about 129,000 ribosomes. And that gave us a 4.2 angstrom consensus map. So it's a really nice high-resolution consensus map of these ribosome particles. And then what we're able to do, because we have the numbers, is we can go in and classify. And so we use a mask. So this is called focus classification, where we put a mask around just the ligands, because the core of the ribosome is the same between every single ribosome across all cells. And so what we do is this focus classification to separate all of the different ligand-bound states. And so when we do that, we're able to kind of separate out and see lots of different states and kind of rebuild kind of the translation cycle or recapitulate the translation cycle as it exists inside the cell. And so what you see here, we don't have all of the different stages of the translation cycle, but you can see we have a few key pieces of it going through. And then we have also this question mark class, which we're not sure. We think it's an incomplete classification, so we're still working on that one. And so then when you map that back into the cell, so we classify based on the ligand states as well as whether it's bound to the membrane or not, and then we map that back into the cell, and you can see here, so this one always takes a little bit of time. So here, now you can see all of that ultrastructure with your ribosomes mapped back in. And so you can see here in bright red, we've highlighted all of the membrane-bound ribosomes, and you can see the localization of those next to the ER makes sense. And then here in the normal kind of this orange color, we've got monosomes, and then here we've got polysomes in this wheat color. And you can see really the localization of the polysomes is kind of clustered around the rough ER. There's much less of them over here next to the nucleus and the digestive vacuole. And so we haven't completely finished this yet, but we're now hoping to do this obviously with the wild type or control cells as well, to see if we see a change in the distribution of polysomes. Are they, are there more polysomes or the less polysomes? Is it more or less translationally active? Is there a change in the number of membrane-bound ribosomes and the localization of the ribosomes as well? And so we're able to go down and kind of do some statistics where we see kind of the difference in the distribution between all of these states, between different life cycle stages. So the ring parasites, the ring stage ones are ex vivo, so these are crudely fractionated, very crudely fractionated, and you can see that there's a massive difference between the crudely fractionated stuff and the stuff that's in situ. So you definitely see more states in situ than you do here. There's kind of much fewer states that you see out here in the ex vivo fraction. And so then we were hoping for a really dramatic change, but what we see instead is kind of a very subtle change, right? And we do see kind of with the 3-hour tropes that the distribution is a little bit different, but we're digging into this a little bit further now. And to kind of help us with this, we're also doing some proteomic mass spec. So we're going in and looking at kind of just broadly kind of the difference in the translation expression of proteins across the different across the different time points. And you can see here that in the first couple hours, there's not that much difference between the wild type, the control and the drug-treated parasites. But as you move further in, so this is a 10-hour and 18-hour time point, you can see that there's a lot of proteins that are being made in the control parasites that your drug-treated parasite is not making. So I like this figure because it shows really clearly that here, this is what a normal parasite looks like, between like the change in expression between 3 and 1 hours versus and then 10 and 18 hours. And here you can see that in the first few hours after drug, the parasite is still managing to make some proteins, but here there's really nothing, right? So at some point, I think that what's happening is that the drug is just really kind of freezing everything and the parasite's just stalling out. It's not making proteins, it's not degrading proteins, it's just hanging out. And so we've done, this is very preliminary, but we've done some geo-ontology of the few proteins that are upregulated, and we see that the majority of them happen to be involved in vesicle-mediated transport of some sort. So this is still this is literally hot off the press. We're like, this data is coming in as we speak. So I don't have a complete story yet, but our idea is basically to go in and eventually have a full story using both the mass spec as well as some polysome profiling, as well as the molecular stuff of exactly what this drug is doing at the molecular level inside the cell and how it's killing these parasites. So with that, I just want to thank all of you for listening, and then thank Hangxun for sending me that crude fractionation of ribosomes in the middle of the pandemic. That really saved me. We had nothing else to image at that time. So that was where the ex vivo ring ribosomes came from. So with that, I'd just like to mention that we are hiring postdocs, and thank you for listening.
Excellent talk. Here, to stage left. Jürgen Bosch. So you showed us your yellow potatoes. So ribosomes are obviously big. What's your limit in size that you could detect? And the real question behind that is, if you take AlphaFold or any other thing of larger complexes, can you start an AI-supported classification?
Yes. Yeah, that's an excellent question. So you're absolutely right that we are limited in terms of size. The ribosome is currently the thing that we're doing because it's the easiest thing to do. I think other things are going to be a lot more challenging. So I would say that things down to maybe 300, 500 kilodalton are still okay. But if you get down smaller than that, especially membrane proteins that are like small and mostly embedded in the membrane, you're going to really have challenges. I think we're going to have to find some way around this really high heme background problem. In answer to your question about AlphaFold, absolutely. I think that if the AlphaFold predictions are good, you can definitely use that for template matching. People are looking into that now about taking kind of predicted structures and using them to create a 3D shape that you can then use as a template for picking out of using kind of machine learning based approaches out of this to varying success. I think that in Plasmodium, unfortunately, the AlphaFold predictions aren't great. So like you sometimes have really great predictions if your protein is highly conserved and very similar to it, how it is in other organisms. But I'm sure that all of you have found that when you go into AlphaFold for looking at your favorite protein, you're just as likely to get a bowl of spaghetti as you are a meaningful structure, right? So I think that at least for our organism, we have to approach that with a little bit of caution.
Hello, Ben Sumner, NIH. The liquid ethane, is that just to avoid the Leidenfrost effect, or is ethane otherwise special?
Sorry, say that one more time.
The liquid ethane?
Yes.
Is that just to stop the Leidenfrost effect when you drop it in from boiling, or is there some other special part of ethane?
Right, so the reason we use liquid ethane is because the specific heat capacity is huge. It's compared to liquid nitrogen. So if you've dipped your hands in liquid nitrogen and it immediately vaporizes, it forms this kind of puff of gas that's much warmer around your hand because the specific heat capacity is very low. And so if you were to do that with your grid, you would have this pocket of slightly warmer air and your grid wouldn't freeze fast enough and you'd get really horrible crystalline ice everywhere. And so the reason for the liquid ethane is because the specific heat capacity is so high that when you plunge freeze it, there's no pocket of gas, it doesn't vaporize, and so your grid really freezes immediately. And you can, if you ever want to test this, don't. But if you want to test this, try sticking your hand into liquid nitrogen and then try sticking your hand into liquid ethane and you'll see like, on the liquid nitrogen, you're perfectly fine. Liquid ethane, massive burns, which I personally have done. So don't do that.
Thank you.
Hi, great talk, beautiful
Thank
work. James McGee, grad student in Scott Williams Lab at Penn State. You and I have had a few conversations about this, so you know I'm going to ask this. So Plasmodium expresses two different types of ribosomes, and of course in asexual blood stage parasites, it's primarily expressing the A-type ribosome.
Right.
When you're going through these different classifications, are there any potential like different structures of ribosomes? Because there is some sequence variation and it's not known if the ribosome protein composition is different. So we're not sure if the structures are going to be different. So this would be a great way to maybe have a hint.
Yeah, for sure. So we, I don't think we're at the resolution to be able to see RNA sequence, unfortunately, quite yet. I'm sure if we keep building, maybe in a couple of years, we'll have enough millions of particles that we can get to that point. But yeah, we don't see anything obvious so far. But I think that definitely potentially going in and looking at ribosomes from sporozoites or gametocytes would be really cool.
Thank you.
Hi. So, I hesitate to ask this question because it may make me look like an idiot, but that has not stopped me previously. Is there any value in looking at things that are fixed by this method? Because there are nice ways, from like Freddie Frischneck's lab and stuff, to get rid of heme that have been done by pathologists for eons.
Yeah.
Right? So you might be able to get really high resolution by getting rid of that heme, albeit you sacrifice. So I guess the question is there value in doing this for fixed cells?
I see.
If there is, there's steps A, B, C, and D to get rid of things.
Right.
But if you never want to look at a fixed cell, then it's pointless.
Unfortunately, I think that the point of this is the high resolution protein reconstructions, and the fixing would ruin that. So I think for ultrastructure, yes, absolutely. The fixing is much less work. You should do that. But I think that for the high resolution protein structures, you're really going to need to avoid fixing.
I mean, the reason I sent you the sample is to looking for mitochondrial ribosomes.
Yes.
In mitochondria, I don't know whether you ever could see it in any of these.
Yeah, so that's exact, yes. So we had a lot of free ribosomes, just like free ribosomes in the sample that you sent us. But unfortunately, we never actually saw anything that we were convinced was a mitochondria in that prep. And the other thing I will say is that, yes, we have hundreds of tomograms now, and we scout every time we see a mitochondria. We kind of really pay extra close attention to that. Also extra close attention to the apicoplasts. And I would love to be able to tell you that we can get a structure that way, but unfortunately, I think I had my students go in and check to count, and I think that the total number of mitochondrial ribosomes we've managed to get out of all of these thousands of tomograms is 10.
Wow, better than
They're
zero.
very sparse. They're
Better
very
than
rare.
zero.
Yeah, and I mean, because you're not even, you're not even, like, our field of U and Z is so small that you're not even catching the entire mitochondria, right? You're catching a slice of it, and then you have to hope that you get ribosomes in that slice. And the same thing is true for the apicoplast. I think the number is maybe 2. So.
Another thing I wanted to do, so since the contrast is low because of hemoglobin,
Yeah.
is it possible just to do sapling lysis? And you don't fix it. You do sapling lysis to remove the hemoglobin, and
Yes.
then put it into this fixation.
So that's definitely a thing that we could consider. We do have some problems with the freezing of parasites that are not intact. They don't pack as well, and so we get a lot of crystalline ice, unfortunately.
But for regular transmission EM, it works very well.
Yeah, so I mean, that's definitely something, that's a great idea. I'm gonna write it down right now, and we're gonna try it. I think the other thing that we would like to do is maybe organelle fractionation, because then you get away from, like, the organelles stay intact, right? So it's still native, but like you don't have that hemoglobin. So yeah, we're actively thinking of everything we possibly can to get around this problem.
Okay.
Thank you so much.
That was great.